This presentation will focus on the regulation of the G1-S checkpoint through the RB protein, cyclin D, and Cdk4 and 6. RB is a 110 kilodalton protein that has 15 phosphorylation sites. One of its major responsibilities is the regulation of the G1-S transition. When the cell receives growth signals, such as TGF-beta, these growth signals activate signal transduction pathways, for example, G protein-coupled pathways, RTK pathways, and RAS pathways that lead to the synthesis of cyclin D along with Cdk4, 6, and P21. During G0, Cdk4 and 6 are inactivated because they're bound to P27. During the G1-S transition, growth hormones trigger RTK pathways that activate IP3 kinase, leading to the phosphorylation of P27 and the detachment of P27 from Cdk4 and 6, which frees Cdk4 and 6 to bind to cyclin D. P21, mentioned earlier, helps the formation of the cyclin D Cdk4 or Cdk6 complex by inhibiting the binding of Cdk2 to cyclin D. The active cyclin D Cdk4-6 complex is necessary for the next step in the G1-S checkpoint, the deactivation of RB. Shown here is how in the absence of growth stimuli, RB prevents transcription by recruiting histone deacetylase, compacting chromatin, and preventing its transcription by E2F family transcription factors. During the G1-S checkpoint, the active cyclin D Cdk4-6 complex phosphorylates the RB protein, causing it to fall off the chromatin and no longer recruit histone deacetylase. This decondenses the chromatin, allowing E2F family transcription factors to interact with the DNA. During G0, RB also prevents transcription by decreasing the affinity of the pre-initiation complex to the TATA box on DNA, preventing transcription. The RB protein also plays an important role in tumor suppression. When there is a double-stranded DNA break, ATM activates the p53 tumor suppressor, which prevents the phosphorylation of RB. This prevents the cell from progressing from G1 to S. Mutations in RB are associated with a number of cancers. Among them are bone, lung, breast, and skin cancer.